Good afternoon, everyone. Thank you so much for joining me. As I was kindly introduced, my name is Shanaz, and I am responsible for the Solentim cell line development portfolio at Nova Biomedical. So today I'm here to talk about how we can strengthen monoclonality assurance in cell line development. So first I'll start with a little introduction about cell line development. So cell line development is the process of generating a stable clonal cell line to produce a biologic of interest. So whether that's the protein that is produced or whether it's the cell line themselves, such as stem cells. As a leader in cell line development automation, Solentim was able to identify three main bottleneck efficiencies. So this includes single cell isolation, proof of clonality, and clone selection. So when it comes to proof of clonality, regulatory authorities expect mammalian cell lines expressing that desired biologic to be derived from a single progenitor cell to be considered monoclonal. So our legacy, Solentim VIPS and Cellmetric, have been widely adopted for their single cell seeding and whole well imaging capabilities. And these have been validated as one of the most effective ways to respond to the specific regulatory requirements on proof of clonality. So here on the right is an example of a paper by our customer Resilience, and they have achieved 100% probability of clonality with our instruments. So the goal of this presentation today is to present the proof of clonality achieved by our newer generation Solentim technology called the VIPS Pro and the Cellmetric X, and that's powered by our Studious software with soon-to-be included fluorescence capabilities. So for those of you that are new to Solentim, the Solentim ecosystem includes a suite of 3 different instruments. So firstly, we have the VIPS Pro, which stands for Verified In Situ Plate Seeding. So this is for your single cell dispensing, and it has imaging capabilities as well. So it actually has all of the imaging capabilities of the Cellmetric X. So the instrument that is in the green background is our Cellmetric X, dedicated purely for imaging. And then the orange instrument in the background is called the Icon, and this is for characterizing our clones. So for productivity, for critical quality attributes such as glycan, and for viable cell density. We also have reagents, which are designed to enhance cell growth at different stages of the cell line development process, whether it's at the clonal stage, whether it's at shaking, or whether you are thawing. So we are really excited to announce that we have just recently launched our chemically defined Instagrow reagent. And this is something that has been requested a lot by our users. So these reagents are designed to enhance cell viability and clonal outgrowth, and being chemically defined eliminates any batch-to-batch variability for predictable performance. And they are animal component-free to meet regulatory compliance. So now let's focus on our new VIPS Pro. So the VIPS Pro performs gentle nanodroplet deposition for single cell seeding. And this is important because you need to have gentle single cell seeding to ensure when it comes to the outgrowth that you have that good colony formation. Other methods can be more high pressure and you end up with single cells, but they don't want to grow. So having gentle single cell seeding is important. It also has high resolution optics for droplet and whole well imaging. And the benefit of this is so you have that visual proof of clonality for that regulatory compliance. Also, having that high quality imaging eliminates any ambiguity. So when you're trying to decide if something is a clone or whether it's debris, making sure you have that high image quality ensures that you make the right decision. We also have the automated media deposition. So once you've seeded the single cell with the instrument, it will also dispense the media as well as part of your liquid handling. And this saves time and reduces any potential error from pipetting. And then we also have our single-use consumables, and this is to reduce any risk of contamination. We also have GMP-compliant versions for your regulatory confidence. So this is basically how the mechanism works when it comes to the VIPS Pro droplet dispensing. So at the beginning, you can see you have the single cell dispensed into a dry multi-well plate. And then your images are acquired, and there is an AI-driven neural network for your clone detection. And then what this will do is it will generate a plate map, and it will show which are your monoclonal wells. So you can see you have all of the green, and that's to tell you these are your monoclonal wells. And then after that, you have your automated media fill. So the AI is a new feature that is not available on the legacy instruments, and this is to help streamline the process of cell detection for the end user. So the VIPS Pro AI neural network has been validated with different cell types. So here I'm showing CHO, HEK, and iPSCs. And there are other cell types as well. So it allows highly accurate detection of over 90% for a single cell in a droplet image. So we have done experiments internally to measure the accuracy against ground truth, so against a human operator. So not only does the VIPS Pro neural network assist the user to detect your clonal wells at day 0, it will also help detect empty wells and non-clonal wells as well. So you can see we have 2 cells there, and it's put a little box around to say this is where we've detected the cells. So now that we've covered the VIPS Pro technology, we will go through the Cellmetric X new generation imager. So there are 3 different versions of the Cellmetric X. So the first one is our basic Cellmetric X imager, which would involve placing a single plate on the instrument at a time. And then we have the Cellmetric X HT, which is our flagship instrument and comes with a plate stacker. So you can automate the imaging of up to 10 plates at a time. And then we have the CMXR, which allows for robotic integration. So the Cellmetric X has your high resolution optics optimized for your whole well imaging. So having those clear images, again, it helps when it comes to that clonality confidence, and it will also do a confluence scan to measure your outgrowth. The plate stacker that I mentioned is temperature controlled, and you can see that is the plate stacker just on the right of the instrument. And having the temperature controlled conditions will make sure your cells are at optimal health during imaging. And then we have the integrated plate handling. So basically inside the plate stacker, it will handle the plates automatically for you. So you can walk away from the instrument and then it will feed the plates inside the stacker. So if you have lots of plates to image, it's going to automate that process for you. And then we have the auto-focusing and the AI-driven analysis. So with the auto-focusing, you don't need to manually adjust the focus. It automatically does that for you. So it's a lot faster when it comes to imaging. And with the AI-driven analysis, it helps reduce any human error and it speeds up that decision-making process. So the Cellmetric X artificial intelligence neural network was trained on thousands of images that were put into the software. And this will assist you after seeding to effectively detect your cell in a well, as well as detecting non-clonal wells and debris. And again, like the VIPS Pro, this artificial intelligence-based neural network is validated on a variety of different cell lines. So here we're showing the CHO, HEC, and iPSCs. And particularly with the iPSCs, you may know that they tend to be like more flat and transparent. But when it comes to the AI network, it can accurately detect these cells. So the second neural network on the Cellmetric X measures the colony outgrowth accurately and efficiently from day one onwards. So it uses whole well imaging with AI, and it's applying a green overlay on the colonies to track that colony growth. And then it will quantify the colony growth and give you a percentage confluence. And it does that up to 14 days. So again, this is to increase that data accuracy, reduce any human error and variability. And it's been trained and validated on diverse cell lines and morphologies. So probability of clonality with the Solentum ecosystem. So internally, we have performed experiments to compare the probability of clonality and Wilson's assurance on our new generation Solentum technology. And then we compared it to data acquired on our Solentum legacy instruments. And what we found is that we had similar results when it came to the probability of clonality and Wilson's assurance. So just to give you a little bit of background about what Wilson's probability of clonality assurance is, if you're not too familiar. So basically this measurement is important because it's based on statistical confidence and it considers the sample size and potential errors. So for example, like if you miss a single cell in a well, and this is stronger for regulatory approvals. So if, for example, you have a large sample size, the confidence interval is narrow and more closer to your observed rate, whereas a smaller sample size, there is more uncertainty because it's just not representative enough. So when it comes to probability of clonality, it actually doesn't take this into account. It only takes into account exactly what you've observed. So when it comes to the Wilson's probability of clonality assurance on the VIPS Pro, we achieved 98.1%, which is good, but it can be improved. So if, for example, you are using only bright field imaging, on day 14, when you have your colony, how can you be absolutely sure that that colony was formed from a single progenitor cell? So there may have been like a ghost cell, additional cell that was missed that formed a colony. So this is where having fluorescence is a very useful tool. So our team is invested in the development of our new fluorescence capability on Studious. Now this is not available yet and it is coming soon. So using our new AI-driven neural network, it will detect a single fluorescent cell in a whole well image on day 0. And this would be for your GFP or RFP tagged cells. And then you have the plate maps, which indicate whether there is a single cell, which is in your light green, aggregate cell locations, which is in your dark green, and then any non-cell locations are shown in white. So here you can see on day 0, again, it's detected a single clone, and then on day 14, you've got that nice colony outgrowth on the fluorescence. So here is an example where on day 0, the AI has detected a single green cell on the left, but by day 14, you can see that a red colony has also appeared too in the whole well image. So the red cell was a ghost cell and was not detected on day 0. And this is why having fluorescence is important in being able to prove your clonality. So using this additional layer of data, having the fluorescence and having the bright field scan, it allows you to achieve 100% probability of clonality with an improved Wilson's assurance. So you can see with the fluorescence, the VIPS Pro fluorescence there at the top, we have increased the Wilson's assurance to 99.98%. from the 98.1% without the fluorescence. Now, you might think that this is a small reduction, a small difference, but it's actually a big risk deduction. So from 1.9% to 0.02%, it's nearly negligible. When it comes to regulatory compliance, you need to have near absolute certainty for monoclonality of proof. And also what this means is that you're going to have a stronger statistical basis for your workflows and to be able to defend those single cell workflows and proving clonality. And then also what this means is that you're going to have fewer repeats, less variability, and faster progress. Now, it's not just a small percentage, it's a 95 times reduction in residual uncertainty, which is huge for high-stakes biologics development. So to summarize, this is the Solentum ecosystem in a nutshell. So we deliver regulatory confidence with 100% monoclonality probability, and the 98.1 Wilson's assurance is boosted to your 99.98% with the fluorescent imaging for workflow validation. So our tools enable robust validation, proving your single cell workflow, and VIPS and Cellmetric are trusted globally for your cell line development. And now with the AI-driven detection, you can save time with identifying clones whilst the Studious software integrates data for smarter and faster decisions. So this is confidence, speed, and innovation all in one platform. So Advanced Instruments is now part of Nova Biomedical. We are here with a full team on booth number 12. So if you want to discover our full range of solutions, we have the Osmotech for osmolality testing and the NovaFlex 2 for bioprocessing. Then please stop by. I'll be more than happy to speak with you. And I would really like to thank you for your attention, and any questions that you have, I will try to answer to the best of my ability. Thank you.